Be recording now. All right. Hello, everyone. I'm showing up on the host list as Asia Davis, but I am actually Diani Figa. I'm the Director of Preservation Services at the Conservation Center for Art and Historic Artifacts. And I want to welcome everyone today to this webinar, which is an introduction to integrated pest management. I'm joined today by Cherry Johnson. I'm going to go ahead and introduce her in just a second. And Cherry and I are more or less going to be switching off throughout the webinar this afternoon, letting you know some information about implementing an integrated pest management program, what to look for at your sites and institutions, etc. But this is part of the Regional Heritage Stewardship Program for Appalachian regions of Kentucky, Ohio, and West Virginia, sponsored by the National Endowment for the Humanities and coordinated through my organization, CCAHA. And we're going to go ahead and get started. So first, I want to just introduce you to Cherry, who's joining us this afternoon. Cherry Johnson has worked in museum collections management and curation for nearly 30 years. She received an MA in anthropology and a certificate of graduate work in museum management from the University of South Carolina, where she participated in a 3-year conservation internship. Cherry has worked with a variety of collections and museums, both large and small, including the South Carolina State Museum, the Florida Museum of Natural History, TELUS, Northwest Georgia Science Museum, Rome Area History Museum, and the Cook Museum of Natural Science. And we're going to be hearing from her in just a moment. Now, we are here today to talk about pests. As always, as in all of our webinars, we're working to try to build a community. So if you've had an experience, please feel free to share by typing in the chat box. We will hold off on, for the most part, answering questions until the end when we have some time built in for questions. So I'm going to hand things over for now to Cherry.
Hi, everybody. Can you hear me? Can you hear me?
I think you're good on sound. Thanks.
Okay, got it. All right, great. Okay, so we're going to start today talking about, I've got a little bit of an echo. All right, so what is integrated pest management? Well, it's a multi-component strategy that is an environmentally sensitive and economical approach to pest control. And primarily that is done through non-chemical means. And I wanted to go back and just touch briefly on the history of IPM, because when I got started as a graduate student and early in my career, I thought IPM was something that museum people had created. But in fact, it's not. It's been around for a long time. And IPM grew out of a dissatisfaction with the purely chemical approaches to insect control back in the 50s and 60s. And it began as an agricultural movement that later spread onto the urban scene. So the problem was that damaging insects were becoming immune to the pesticides. Their natural insect predators were being killed off, and so the insects were coming back in greater numbers. And then in 1962, Rachel Carson published her famous work, Silent Spring, and got everybody thinking about the fact that pesticides are extremely dangerous to the environment and to us. And so today, IPM is used in a lot of different industries, in schools and hospitals, and of course in museums and libraries. And again, it's a holistic approach that aims to manage pests through blocking, climate control, monitoring, eliminating a variety of attractions, and establishing good housekeeping procedures. All right, so let's go to the next slide. Why use IPM in cultural heritage organizations? Well, there are many reasons to use IPM. We want to control and eradicate pests that attack and damage our collections and our records. And we would prefer to use a method that doesn't use pesticide, because these are less harmful methods to humans and the environment. IPM is also more cost effective. It's easier to prevent, and it's more sustainable than pesticide use. And finally, it's safer for the objects in our collection, the ones that we're trying to protect. We know that pesticides can cause physical damage to objects, things like altering color, damaging plastics. They can easily be absorbed into some materials, and that can be potentially irreversible. And then in the case of natural science specimens, some pesticides can rapidly damage the cellular material, and that makes them useless for DNA studies. Okay.
So IPM can be broken down into a four-pronged approach: avoiding and blocking pests from establishing a home in your collection, detecting an infestation, which is usually achieved by monitoring and then assessing the extent of the situation, identifying exactly what the pests in your collection are and where they are, and responding in a way which is sensitive to the needs of the collection, staff, and the environment.
Okay, so avoid block. This is the first step we want to talk about in integrated pest management, because if your pest can't get in to begin with, you're not going to have a huge problem. I kind of like to liken the museum environment to a set of nesting dolls, the ones you see here. So first you start with your building envelope as the primary defense. So pay attention to the foundation, the walls, the roof, the windows, and the doors. Insects can get into tiny spaces, in through tiny spaces like 1/64 inch, maybe even smaller than that. Rats through an opening the size of a quarter, and mice through a space the size of a dime. So then inside your building you have your collections room or even your exhibit hall, and then inside of these rooms are cabinets or shelves, and then finally in the cabinets you may have individual boxes or containers. So there are lots of levels where you can protect and block, and you really should pay attention to each of these. What attracts pests? Well, the four biggies are food, warmth, humidity, and hiding spots. Removing the things that attract pests is an important line of defense for your institution. Pets are drawn to collections for these four reasons. They're attracted to food sources like paper, wood, leather, wool, human food, dust, feathers, and actually other insects. They are also attracted to warmth, so 68 degrees and above is really prime climate for insects. They're attracted to high humidity and dampness, and then also a lot of the little hiding spots like ventilation ducts, cracks between the floorboards, flowers and plants, dark places, old boxes and piles of paper, and I'm going to even add firewood. If you're in a historic home, you may have stacks of firewood. In terms of humidity, if you can keep your humidity down to at least 50, but even a little bit lower to 45%, you're going to eliminate problems with socias or the book lice, because these live off of mold and mildew and they retain water in their bodies. So if you can eliminate or keep the humidity down, then they'll just pretty much dry up and die. Okay, Deani, are you going to take this slide, or would you like me to talk about that one?
I can. So regular routine housekeeping is potentially one of the most important things you can do to avoid a pest infestation. It removes food sources for the pests, and regular cleaning also allows you to monitor some of the dark or hidden areas in your collection for nests. Having time to regularly and thoroughly inspect your collection for early signs of an infestation is crucial to stopping a problem in its tracks. Sealing gaps in the building envelope and repairing holes and cracks will also help block pests from coming in. It sounds really simple, but it can be highly, highly effective. Also, extremely important is segregating potentially infested objects from other collections materials as they come in. That can be very crucial in controlling an issue. This can be done using a procedure known as isolation, which we'll cover in a little bit. Being vigilant about things that enter the collection can save you from a lot of difficulty later on. You know, ideally, an institution would quarantine everything for up to a couple of weeks before it goes into the permanent collection storage room. Not every institution, or maybe even many institutions, have the space for that, but it is something to consider. So much in the way you would prepare for potential security risks or natural disasters, it really helps to evaluate your institution for potential pest risks. This means doing a thorough inspection, conducting a monitoring program, and getting to know the creatures that may inhabit your site. The first series of questions you want to be able to answer before there's a problem are some of the ones that are up here. So which parts of your collection are most at risk? Is it, for example, the herbarium specimens or the collection of rare books or both or something else entirely? Which part of the building or buildings are most at risk? Are there activities which your institution carries out regularly, such as serving food and drink during events, which might present a good opportunity for a pest infestation? And are there signs of insects or other pests that you or your colleagues have noticed in the past? If you do believe that you have a pest infestation, then the next step is to evaluate how serious the situation is. Where have pests been noted in your collection? What stage in the life cycle are they? If you found larvae, that indicates that they're probably breeding, for example. Is there any visible damage? Are you finding carcasses, or are they still alive? How many are they? How many are you finding, and of what species? And the really scary and, getting to be one of the most concerning questions, how many objects appear to be affected? We will discuss the materials that attract pests when we discuss identification, but just in general, pests tend to be drawn to proteinaceous materials such as hair or leather and vegetable matter or starch. So unfortunately, that just about covers a large majority of the materials found in cultural historic collections. Pests particularly like these objects if they are dirty or damp because that provides extra food and hydration. If you're in a historic house, then your largest artifact, the building itself, can also definitely be at risk. Of course, termites love wood. So the next phase then is monitoring. Monitoring insect populations with traps is simple and effective for pinpointing exactly what and where a pest infestation occurs. So this isn't the part where we're actually stopping, physically dealing with a pest infestation, but it's really learning more about what's happening at our site and where. Sticky traps, or blunder traps as they are also known, are probably the most common type of trap used to monitor insects. Blunder traps, of course, is because the insects are going to be just kind of strolling along and then blunder across the surface and get stuck in there. They're designed to trap crawling insects. These are small enough to be placed in dark corners and underneath furniture, so they can be relatively out of view. They may also trap things like lizards and mice. So we'll discuss humane traps in a minute, but it is good to know that you can break down the adhesive and free these types of pests if you'd like by using mineral oil. So if something gets in one of these other than an insect and you want to set it free if you discover it in time, there are ways that you can deal with that. So those were for crawling insects. Pheromone traps can be used for flying insects, such as moths, or some crawling insects, such as roaches. If you're looking into this option, it's important to know what types of pests you're trying to trap, as the pheromones are going to differ for different types of insects. Male insects are drawn by the hormone given off by female insects to attract a mate. Those are known as pheromones. And in these traps, they're held in small vials within the trap. Once the insect enters the trap, they cannot get out, which cuts down insect breeding and helps to track their movements. Traps should be placed in a regular pattern around the building with their positions marked on a map of the building or area of the building you're trying to track. This is an example of a pest monitoring log that you might be able to adapt and use at your institution. This is one that came from the Utah State University. Traps should be inspected regularly with the number and type of insect recorded on a similar log, such as this one here. This will help you to determine whether the insects you're catching are actually pests, which we'll talk a little bit more about in a moment. And it will also help to determine the life cycle. If you notice larvae in the trap, for example, that indicates that the insects are in their breeding phase. For non-insect pests, the same procedure applies, although you may want to check them more frequently because if it's left too long, it's possible that an animal carcass may attract additional pests. Traps don't necessarily have to kill. For example, humane traps are available for mice and other rodents. You must check and empty them regularly, though, and you must take them to a place relatively far from your institution or those pests will come right back in. This green house with mice in it is a type of humane trap called a smart mouse trap, and they're available online and at most hardware stores. So in conclusion, traps are necessary to identify and track pests and non-pests, pinpoint an increase in insect population in a specific area, monitoring the spread of pests from one area to another, tracking an invasion of adult insects in the summer, for example, pinpointing a localized infestation in a problem area, and determining the success or failure of a control treatment you may have recently implemented. Cherry now is going to talk a little more about identification.
Okay, great. So figuring out what pests are in your collection can often seem really overwhelming. Insects can look the same to the unaided eye, and that can make identifying them a little tricky. So let's begin by looking at how you can identify the various insects and other pests which might be in your collection, because knowing what signs to look for can help you to nip a growing problem in the bud. So this is a map of your area, and you'll see on there some different insects that actually may be in your area, but really we have a lot of the same pests across all of the United States, although the species may vary. And these typically include book lice, cigarette beetles, carpet beetles, cockroaches, termites, case-bearing clothes moths, silverfish, and fire brats. And there are, of course, many others, but these are some of the most reliable offenders that we have. And when we talk about lovely hiding space for things, I want you to take a look at this little guy right, here. Down here at the bottom of the page, this is the brown recluse, and they're common across North America, but here in Alabama where I am, we see them a lot. They love to hide in cardboard boxes, as do black widows, and they may not hurt your collections, but they can hurt you. So just be aware. I know a lot of sites in our storage areas, we may have boxes that have been sitting there for a while. Just be vigilant about looking for them. There are a lot of common myths that brown recluse will kill you instantly. That's not true. Actually, death by brown recluse is really rare, but just be cautious about it. Okay. Know your pest. First of all, what is a pest? Well, for our purposes, a pest is an insect or other arthropod, rodents, birds, or really any other creature, any creatures who don't belong inside your building. And pests can also include fungus and mold. So in terms of pests, we have vertebrates, which can vary by region, and common vertebrates include rodents like rats, mice, and squirrels, but also birds, and also occasionally some larger vertebrates like rabbits, feral cats, rats, raccoons, possums, and snakes. You're less likely to see these larger vertebrates in your collection, but they are more common than you think, and especially in historic homes. So there are also the insects and other arthropods. So think, in addition to insects, the spiders and scorpions. And then there's the fungus and mold, which indicates you've got a humidity problem. Can really ruin your collection, and also it serves this serves as food source for other pests. Okay. So these are some signs of pest activity. Droppings, gnaw marks, nests made from shredded paper and cloth, paw prints, noises, and unusual odors. And then for birds, you may see nest, feathers, urine, and excrement. And let's see. Let's get rid of this little arrow. We don't need that anymore. Just be careful when you are cleaning up the urine and excrement to wear gloves and masks, because some pretty nasty diseases can be associated with that. So again, just be mindful of that as you're working. Okay, Diani?
So signs of pest activity for insects are less obvious, although certainly much more common. For the purpose of identifying insect pest activity, we'll divide them into three general categories of destruction: borers, shredders, and grazers, and I'm going to talk about all of those in a little more depth. I encourage you to use one of the online pest atlases to visually identify pests and the damage they leave behind. Towards the end of this presentation, I'm going to have a slide with a link to a bunch of different really excellent resources for finding out more online, and I would highly encourage you to consult those about specifics for your collection or for your site. There are so many insects out there, and often they can look very similar to the human eye. But there is definitely a wealth of information online which is constantly being updated. So, getting into a little more details about those three broad types of insect arthropod pests. Borers. These types of pests damage hardwoods and softwoods, animal glued plywood, furniture, wicker, wood pulp paper, and books. They leave behind exit holes, tunnels, and frass, which is basically the garbage left behind by the insect larvae. If you see these holes and brush the inside gently, powdery frass may come out, which is a sure sign that an insect has been breeding. The frass itself can also leave behind stains or even damage the object. Borers include the common furniture beetle, the woodworm, which is actually the larval stage of the furniture beetle, and the powder post beetle. The powder post beetle prefers hardwoods such as oak, ash, hardwood, and hickory. They can be found in the envelope of your building as well as in wood objects within the collection, such as furniture. Common furniture beetles and their larvae, which I mentioned are also known as woodworm, enjoy damp wood environments and areas with high relative humidity. As an aside, most of these insects are tiny, really only a few millimeters long. This is where it definitely comes in handy to have some type of magnification device to be able to differentiate various species from one another. Next up are shredders. These are probably my personal least favorite type of pest. They just make me so angry, destroying my clothing. Shredders consume keratin, which is the protein in hair, wool, parchment, feathers, skin, horn, nails, and hooves. They are the enemies of many textile and natural history collections, and they can leave behind a mess of cast-off skins, silken tubes, and frass. Shredders include the webbing clothes moth, case-bearing clothes moths, and the white-shouldered house moth. So these, as I hinted, if you have ever found holes in your favorite sweater or other wool or other textile materials, you most likely have webbing clothes moths in your home or site. Sometimes these moths are confused with the grain-eating moths you might find in cupboards at home, but clothes moths have tiny hairs on their head. So if you catch a clothes moth and look at it under magnification, you should be able to see that they have furry heads, which you can kind of see on that image up there on the screen. The larval stage is actually the point that is the most dangerous for collections, as the baby moths feed on protein as they grow. By the time you see them fluttering around, they have probably already done some damage, and they are ready to breathe and start that cycle all over again. White-shouldered house moths are actually omnivorous, so they are attracted to grain sources as well as the protein and wool, fur, etc. They are easy to identify, at least under magnification, by the white patches where their shoulders would be if they had shoulders. Many species of beetle also fall into the shredder category. Varied carpet beetles, which actually kind of look a bit like a decorative carpet themselves, those prefer natural history specimens, rugs, textiles, fur, other insect specimens, and furniture. Their larvae are fuzzy and look like tiny caterpillars. The black carpet beetle enjoys both animal protein and plant-based materials, so those ones are also omnivorous. The vodka beetle, not nearly as fun as it sounds, prefers protein-based materials such as wool, skin, and hair. They are also attracted to dried herbarium specimens and other plant matter as well. And then finally, the drugstore beetle is also known sometimes as the bread or the biscuit beetle, and it can be found in dried vegetative material as well as furniture, natural history collections, fur, leather, and books. Moving on are the grazers. These pests primarily feed on starch and protein and thrive in damp conditions. So these are definitely some of the ones in paper-based collections that you might want to be the most concerned about. And actually, a great deal of pests thrive in high humidity, as Cherry mentioned. So keeping the relative humidity in your collection controlled is crucial in avoiding an infestation. Grazers damage objects by scratching and eroding them, as you can see in this image here, in which silverfish have very, very badly damaged a book. Grazers include common household insects such as silverfish and cockroaches, as well as book lice and cigarette beetles. Cigarette beetles infest books, herbarium specimens, leather, silk, and natural history objects. They tend to become very active in subdued light and fly around and then play dead when the lights are on. Book lice are not actually lice, but they have a similar appearance. They feed on fungi and mold, so if you see book lice present, that could indicate that you have a mold problem as well. At the very least, it probably indicates that the relative humidity is too high, or at the very, very least that you want to look into your relative humidity situation. Book lice are drawn to the starchy adhesives in wallpaper or books. Lowering the relative humidity in an affected area to 50% or below will prevent their development by eliminating potential moldy food sources. And then silverfish, which many of you are probably familiar with, are attracted to starchy adhesives as well as proteins. So this means that they will eat book bindings, carpet, textiles, glue, hair, paper, photographic materials, cotton, silk, and linen. You can create an inexpensive kit for identifying pests, assembling materials that can be found almost anywhere. So for somewhat of a starter Pest ID kit, you might want to gather the following: a microscope, magnifying glass, loop, or some other type of magnifying device. We talked about the importance of being able to see particularly those insect specimens up very close and magnified. And you can get a microscope that is actually, you can plug it in with a USB. So those can be quite manageable. A pest atlas, such as one of the online resources that we're going to be looking at a slide for in just a moment, or a pest identification book with good photographic images. Sticky traps or insect food pheromones. A light source, such as a flashlight, ideally even an LED flashlight that you're not going to have to worry about changing the batteries on a routine basis. Tweezers, small Ziploc baggies or plastic or glass vials with lids for collecting specimens, a small brush, a pencil, your pest logbook, and a camera for documenting images. Now this is a bit of an aside, but I do want to just give kind of a shout out that to something which is occasionally overlooked when discussing insect pests, which is the reminder that not all insects which you might see at your site or even around collections are necessarily pests. So it's important to differentiate insect pests from insects or other arthropods writ large that might, appear gross or appear like a nuisance but aren't going to damage your collections. A coworker of mine mentioned that she heard from a pest management specialist recently that if you find absolutely no insects at all at or immediately around your institution, it probably actually means that the environment is toxic and you don't want to be there, which if you think about it makes a little bit of sense. One type of creature which is particularly helpful for pest control is it's an arachnid that is the pseudoscorpion or book scorpion. They're so small that if you see them, you would probably overlook them in your collection, but they are actually team players when it comes to pest management. They eat some of the insects which you don't want in your collection, such as book lice and clothes moths, while leaving the books themselves and other paper materials alone. Think of them as, unpaid interns, volunteers who are just very unassuming and quiet. But I think it is important to acknowledge that non-pest insects are important in that they can indicate another problem at your institution, such as building or environmental issues. So if you are seeing a great deal of insects inside of your building, they might not be insects that you're particularly concerned about for damaging your collections, but you might want to go ahead and check on your window seals, check on the foundation in your basement, see where those points of infiltration might be. That might be a sign that triggers something else. So monitoring will help you to begin evaluating whether or not the insects in your collection are, in fact, pests. Now, Cherry is going to talk to you a bit about response.
Okay. There are many different options for responding to an infestation, but we're going to briefly touch on four options that are used by institutions. These include isolation, low temperature treatment or freezing, heat treatment, modified atmosphere treatment, which includes anoxic carbon dioxide and nitrogen argon gas treatment. And then we also talk about two options that were common in the past, fumigation and pesticide treatment, that really aren't recommended now. It's helpful to know about all these options so that you can make an informed choice, particularly when you're working with a pest control company or a professional who may not be necessarily familiar with the best treatments for archival and library objects or three-dimensional objects. So just as a reminder too, whatever you do and whichever treatment you choose, always document in detail what you find and how you're responding to it. So let's talk about the first one, which is isolation or bagging. An object suspected of infestation is placed on a white backdrop, so a blotter or some other white archivally safe material, and then sealed in a polyethylene bag. The sealed package, you're going to then monitor it for several weeks or even months. This is especially good for objects that cannot, for whatever reason, be frozen or treated right away. The pros of isolation are that it allows you to quarantine an object that may be infested, that you suspect is infested, so that nearby objects are not affected by that. So you want to get it away from your other objects. It also allows the object to be monitored for weeks or months so that pests can be identified and the life cycle stage determined, because some pests are not harmful in certain stages, but only in other stages. And they're also more difficult to terminate in certain stages. So it really just lets you monitor that process. Isolation can be used with a lot of different materials. It's appropriate for complex objects and objects entering or re-entering an institution. So new items to your collection or items that you've sent out maybe on loan that are coming back in, you can use isolation and bagging just to make sure they're not having any problems. And I would actually suggest that if you have the space, you always want to quarantine new objects coming into your institution. Now, a lot of institutions don't have that space, but if you do, it's something you might consider. So the cons of isolation and bagging are that it's time consuming, it takes weeks or maybe months, and it's only appropriate for a small number of objects. The next option we are going to talk about is low treatment temperature or freezing of an infested object to kill the pest. It's sometimes used as a preventive measure when items arrive from a place where you suspect there's an infestation. The first thing you do is place the object in a polyethylene bag. If it's too large for a bag that you can purchase commercially, you can use polyethylene sheets and make your own bags. Then you want to put the objects in the freezer for a specified time length, and that depends on the capacity of your freezer. But it should be at least 72 hours. When you put the objects in the freezer, make sure they're not touching, and you want the air to be able to circulate around them. And also you want to include an indoor-outdoor thermometer so that you can monitor the temperature in there. What you want to do is to make sure the objects get down to at least, the temperature gets down to at least -20 Celsius or -5. degrees Fahrenheit. That's the minimum low temperature. Um, So a lot of studies have been done on this lately, and in recent conversations that I've had with pest control specialists and some conservatives, I'm conservators, sorry, what I'm hearing is that and what I did is you can actually use a household chest freezer to do this treatment. And you can find these that get down to the minimum temperature that you need. It's really important that if you go this route, you choose a freezer that does not have a frost-free cycle. You don't want it lowering the temperature and cleaning out the frost, because it interrupts the consistency of the temperature, and that's really important to this process. So if you purchase a household chest freezer to use and you get it down to the temperature you need, the formula is 6 to 10 days at -20 Celsius. If you can't get it down that low, you can leave it in for longer, say 2 weeks. Sometimes you may want to repeat the freezing cycle, particularly if you're dealing with wood-boring insects. And if you do 2 cycles, so what you're going to do, it's similar to doing 1 cycle. You freeze it, you take it out, but leave it in the bag for 24 hours so that it can come down to room temperature. And then if you want to put it in for another cycle, then you do it at that time. If you're only doing the 1 freezing cycle, you can leave the objects in their bags and put them into collection storage that way. And then remember always to document this process. So the pros of low temperature treatment are it's a short treatment time, but it can't be used on a variety of objects. So you can't use it on different finishes, oil or acrylic paintings on canvas, objects with layers of media, photographic materials, cased photographs, glass archival materials, ceramics, metal, glass objects, or objects under tension like a drum. You also don't want to use it on ivory or teeth. And then the low temperature treatment can increase the fragility of delicate objects, especially as you return them to room temperature. Okay.
So I'm going to talk about heat treatment now. Exposure to high temperatures can be used to kill insects as well. Short exposures of about 130 degrees Fahrenheit are sufficient to eradicate many pests, and this treatment can be scaled from using a chamber for a few objects on up to a large object on site, or even an entire building. There are various versions of this heat treatment. 2 proprietary names for heat treatment are thermolignum and solar bagging, which uses the sun to reach the high temperatures needed to eradicate pests. Pros of heat treatment are that it is very effective. It's when you get up to temperatures that high, it really does the job. It also is a pro that, as I mentioned, it can be applied to both large and small scale infestations. Again, from just 1 object to an entire building. Cons of high heat treatment are that it's not recommended for items which are particularly sensitive to changes in relative humidity. Temperature increases the ability of the air to hold humidity, so it can cause swings in relative humidity. So it's not just the temperature that might be subject to change. The RH can definitely have a significant fluctuation there as well. It's also not recommended for what are probably fairly obvious reasons for collections which contain low melting point waxes, as well as some adhesives, certainly flammable and explosive objects, and then of course some plastics which melt or deform when exposed to heat. Next, we're going to talk about modified atmosphere treatments, including anoxia, controlled atmosphere treatment with carbon dioxide, and nitrogen or argon gas treatment. So an anoxic treatment often uses oxygen scavenger packets, which pull the oxygen from the air. Anoxic means without oxygen. The infested object or objects are placed in a sealed enclosure with those oxygen scavenger packets, and then the air is removed. Oxygen levels are then reduced to less than 0.5% for 3 weeks, which results in an atmosphere which is almost entirely composed of nitrogen. The treatment length necessary will vary, but approximately 21 days, that 3-week period, is sufficient to kill almost all insects at all stages of life. Pros of anoxic treatment relative to other modified atmosphere treatments, it's pretty inexpensive. It works for a variety of materials, and it tends to be very effective. Cons are that it's time consuming. I mean, you have to wait those approximately 3 weeks until there are trustworthy results. It can be difficult to maintain the relative humidity equilibrium in that type of environment. It must be monitored. You can't just kind of set it and forget it. You have to, and for 3 weeks, there's a lot of monitoring that is entailed there. And you cannot use this type of treatment on certain objects. For example, those objects that contain Prussian blue dyes or pigments. Next, we're going to talk about a controlled atmosphere treatment, abbreviated to CAT, but it's obviously not the feline that the image up here indicates. This is often inaccurately referred to as an anoxic treatment, so it is actually different. A CAT treatment uses carbon dioxide to displace oxygen in a sealed enclosure to levels low enough to kill insects at all stages of life. After, again, about 3 to 4 weeks, the carbon dioxide is then removed and the treatment is completed. Pros here are that it's safe for most objects. You can actually just set it up in-house. It's very effective at killing insects from all stages of life. There are little to no residual effects on most collections materials, so that goes right along with that first point about it being safe for most types of materials. Cons are, again, it's time consuming. This one is expensive. It requires highly specialized equipment, knowledge, and possibly permits or licenses to use. So all that you're kind of gaining and being able to take this on in-house might be countered by the fact that it's pricey and you're going to have to bring in specialists. This one also definitely must be monitored throughout the process. And then finally, in terms of these the modified atmosphere treatments, we're going to be talking about nitrogen or argon gas treatment. This one works similarly to other modified atmosphere treatments, using nitrogen and argon gas to push oxygen levels down to levels inhospitable for insects. Insect death in these types of treatments usually result in between 2 to 6 weeks, so it can be a bit quicker than the other ones, but it can actually be a bit longer as well, so the range there is just wider. So the pros here are that it can be, although not always, more time efficient than the CO2 treatment. This one also is appropriate for a fairly wide variety of objects. The argon can prevent biodeterioration from fungi and bacteria. Cons here are that this one too is also pricey. It's again highly specialized and significantly more expensive than some other options. And the nitrogen, although the argon is preventing the biodeterioration from fungi and bacteria, the nitrogen can actually contribute to the growth of certain microorganisms. So finally here we're going to talk about a few techniques or practices that are not recommended overall, with a few caveats, but just explain some kind of frequently asked questions about. We're going to talk about first fumigation and then just pesticides in general. So fumigation was a standard treatment in collections for a very long time. And unfortunately, some objects in museum, library, archival, and other historic or cultural collections still have residue from this type of procedure. It is not recommended anymore, except in cases where it's an absolute last resort. If fumigation is chosen, if you have such a severe pest infestation that for whatever reason it's determined that's the only route you're going to be able to go, you will want to remove the object from your collection and send it out to a facility, as you really do not want to fumigate your entire site or collection in situ. Chemicals which were considered safe in the past have actually left dangerous residues in some objects, including arsenic and mercury. A coworker of mine recounts a survey she conducted at a library where they had several books that had actually been painted with mercury or a mercury compound, and the residue from the treatment was still visible many, many years later. One final note about pesticides. We've talked a lot about how to avoid them by implementing this integrated pest management program. And it is worth, though, even though we've mentioned it before, it's worth reiterating that you never ever want to treat collections materials directly with pesticides. Treating collections materials directly with pesticides can change not only the physical structure, but even the chemical makeup of an object. And as I just discussed in talking about fumigation, it can even also leave a possibly toxic residue. It's actually illegal in some states for anyone who is not a certified commercial applicator to use large quantities of pesticides. So any use of pesticides would need to be done through a licensed and registered vendor. And then, of course, with that decision, hopefully at your site, you would be able to have the opportunity to have collection staff weighing in on, just where and how those pesticides are applied. Buildings can be treated in some cases, particularly in the cracks and crevices of the building envelope, which is, as many of you probably experienced, quite common, but only by licensed individuals. And ideally, if you are going to go that route in terms of treating a building with pesticides, ideally you would want to hire a contractor who is familiar with treating museums, archives, libraries, historic sites, and again, other cultural or historic collecting institutions. So Cherry is going to talk briefly about creating an IPM policy.
Okay, thank you. It's really very important to have a written policy that you can share with your staff, your board of directors, and your volunteers. Really anybody. So IPM is the responsibility of the entire staff and your stakeholders. As we say here, if it is not in writing, it doesn't exist. Your IPM policy really needs to be one of your core documents for your institution. And it ensures that all staff members know what to look for and what to do, so that when a staff member or even a volunteer sees an insect, they'll know exactly how to handle that situation, because one of the last things you want to do is have them step on it and squish it, because then you can't identify it. So it includes educating volunteers and staff, and it standardizes procedures. Additionally, an IPM policy helps you in collection management decisions. It ensures consistency with other policies in your organization. It establishes a foundation for communication with other departments like facilities management and custodial services. And that's really important because whoever is cleaning your building or making out the plans for how the building is going to be cleaned need to be involved in your IPM plan. So that you will have a swift and unified response to any infestation or to just to how you're going to manage pests within your institution. So get it in writing, make sure everybody is on board, and make sure it's consistent with your other policies.
All right, so we are just about at the end now, and I just want to thank you all for joining us in this webinar. We definitely have plenty time for questions, so if you have questions, Cherry and I will be happy to answer them in a few moments. If you want to go ahead and start thinking of your questions and starting to type them in the chat box there. I know that speaking for myself personally, and I'm pretty sure this is the same for Cherry, if you want to email us later, that's definitely totally fine as well. Already got one question, will the slides be made available? So the entire presentation is recorded, and the recording of the presentation will be linked through our website. There's in the RHSP section of our website, there's a whole list of previously recorded webinars, so it'll be accessible there. If there are particular slides that you want the text only from that one particular slide, go ahead and shoot me an email and I can send that along your way as well. But that's actually a good lead into one of the main things that in terms of sending out resources, folks are often interested in, and that's those online resources for pest identification that I mentioned a few times earlier. As I mentioned, having a visual aid can be very, very helpful. So these sites all have either images of pests or ways to even submit a photograph of a pest in your collection to be identified if you want help from specialists. And so the four up here, the three bottom ones are really great resources for pest identification and information in general. The top one, museumpests.net, is specific to museums and other cultural institutions. And that's one that I mentioned is updated quite frequently with changing information if there's new and different information about insects and other different types of pests. So I'll go ahead and leave this resource slide up for another minute or two before I flash forward to our contact information. But let us know other questions you have. Go ahead and type in the chat box, or if anyone wants to share, problems or concerns you've had at your institution, we could commiserate together on that. So I'll give you a moment to think those over. While you're thinking, while you're typing, I'll just let everyone know that we have our January webinars scheduled. One of the Conservation Center's photograph conservators is actually going to be speaking about an introduction to photographic preservation and conservation. So photographic materials having certainly in many cases very unique and specialized issues with their preservation, housing, and other care. So she's going to be talking about some of those unique examples, specialized formats.
Looks like we've got a couple of questions in process here.
Yeah. So, I have a one thought for Mary, and then Cherry, maybe you can add to my thought. Mary had a question. You acknowledge that not every institution has a separate area to isolate new collections. Are there any recommendations you would have when you can't segregate a new collection to help protect other collections? One thing that you can do, and this is also certainly space contingent, and you can't, if you get a really massive collection, you can't necessarily do this all at once, but sometimes institutions have it as part of their acquisition protocol to just kind of preemptively freeze everything before they integrate materials into their permanent collections space. Just in case it doesn't even, it buys you time that you don't even have to sort through everything to determine if there are concerns or problems, but it's just kind of a better safe than sorry approach. I wouldn't necessarily recommend that for everyone, but that is one way that you can. Certainly a chest freezer takes up less space than having an entire segregated room where you can spread things out.
Right. Just keep in mind that some things can't be frozen. That would be the only
Sure.
drawback. But yeah, I think that's can be a good practice to do. Go ahead and freeze everything if you're able to do that. And if it's a small collection that you've gotten in, then bag it and isolate it on a, as long as you've got it bagged or even double bagged. That's one way of quarantining it.
Absolutely. My one, just to be kind of sort of a devil's advocate to that, but my one caveat with that is that I visited a lot of sites who do the preemptive bagging as isolation, and I definitely don't think there's anything wrong with that as part of a protocol, but I find that it's easy to kind of just never deal with the materials then. It's a really easy way to kind of forget about your unprocessed acquisitions and just have things that are bagged on the shelf for what starts out as days and ends up as years. So just, if that's part of your protocol, just stay on top of it. Let's see. How often would you recommend checking the sticky traps?
I would check once a week.
Mhm.
I don't know how you feel about that, Tiffany.
I think that would be certainly ideal. Yeah, for sure. Uh, at the very, very, very least, I would definitely want you to do it more frequently than this. But one thing I'll say is you want to really be sure to stay on top of monitoring when the seasons are changing, because there might be very different types of pests, obviously, at different times of the year. And so, I would definitely want you to check your sticky traps as often as once a week if possible, but maybe have a quarterly plan for going through, if you're keeping pest logs, for really going through everything with a somewhat more fine-tooth comb.
And the funny thing about the sticky traps is you can forget where you put them, so it's helpful to make a map of where they're located in your building.
Yeah. Box elder bugs in the archives. From what I can tell, these are not pests. Just want to clarify if you know if this is common and if I should be worried about them. Adult bugs, and they tend to die quickly on their own and haven't damaged anything yet. I do not know of them. I think, to my knowledge, you're correct. They're not a major concern for collections. Cherry, certainly feel free to jump in. One thing that I will say about something that you wrote is if they tend to die quickly on their own. So if you're finding their carcasses, just make sure that you're cleaning those up frequently, because certainly insect carcasses can be an attractant to other types of pests that might be a concern for your
In-
collection. So,
Insects love to eat other insects, for
mhm,
sure.
mhm. Good. Any other questions or thoughts? Like I said, you can definitely reach out to me with any questions. Here on this final slide here are both of our email addresses, and this URL here links directly to information about the Regional Heritage Stewardship Program overall, which is where you can further find those webinar recording links. And this one will be up in probably a few days from now. Well, I don't see anyone else typing, so I'm going to maybe go ahead and call it. Thank you everyone so much for joining us this afternoon and learning about pests and hearing about some potentially kind of gross issues with that, I hope for all of your sake you never have to deal with. Happy New Year, pest free. Yes, I hope that for all of you. So thanks so much for joining us in our final webinar of 2017. Thanks very, very much from CCHA and RHSP2 Cherry, who contributed.
Thank you.
Oh,
And
of
if y'all
course.
think of questions later on, please, because we know sometimes that's the way it works. Later on, you think of a great question, so do email us.
Yes, please do. And please join us if you're interested in January for information about photographic preservation. We'll be sending out the exact dates and information on that shortly. So thanks, everyone. Have a great rest of your Monday.
Happy holidays.
Happy holidays, everyone. Thanks.